Good day, everyone. My name is Katharina Hartmann, and I am the Global Head of Vaccine Safety at Takeda Vaccines. The topic of my presentation today is Vaccine and Drug Specifics in Good Vigilance Practice. Just to start with the disclaimer, this presentation reflects only the authors, meaning my views, and the Union is not liable for any use that may be made with the information contained herein. This lecture is part of the Module 10 Vaccines on the outcome measuring effects of vaccines in pediatrics. And the goal of this lecture is to show the contrast between vaccine and drug specifics in good pharmacovigilance practice based on the new European legislation. Please let me start with some safety aspects specific to prophylactic vaccines. Vaccines are administered to healthy people, including infants, neonates, elderly, geriatric population, and immunization is even mandatory in some countries. Adverse effects are less acceptable for vaccines as benefits are not immediately visible as they are for drugs. So there is a lower risk tolerance. Intensive investigation of rare and serious adverse events become necessary, but such investigations are rarely conclusive. The age of vaccinees may coincide with the emergence of age-related diseases, so neurodevelopmental disorders may occur in the first year of life when infants get quite a number of different antigens. Vaccines are complex biologicals, which makes assessment of causality difficult. They are highly complex multi-component products. They are manufactured from biological systems which inherently vary over time and between the manufacturers. Therefore, brand and batch-specific pharmacovigilance activities due to these biological variability become necessary. Also, the benefit-risk balance depends on factors acting on the population level. Vaccines provide community benefit, so-called herd immunity, and not only individual benefit. The benefit-risk balance becomes dynamic and may change over time. And changes may include efficacy and effectiveness in vaccination programs. Therefore, safety concerns may have an impact on vaccination programs, with potential loss of confidence in immunization programs that may have a significant negative impact on public health. And therefore, effective communication about vaccine and vaccination safety becomes necessary and is also difficult, as perception of harm may persist despite evidence that the serious event is not related to the vaccination. So vaccines are biologicals, as I already said, and variability and changes in the manufacturing process may have an impact on quality, efficacy, and safety, and therefore pharmacovigilance should always be informed on changes in the manufacturing process. Safety considerations of different types of vaccines are important. So there are differences for life-attenuated vaccines, for inactivated vaccines where we have bacterial proteins, we have polysaccharides, we have protein-conjugated polysaccharides, we have recombinant vaccines. We also have new vaccine concepts with new technology and new delivery systems. We have vaccines with new adjuvants, alternative routes of administration, and we also have the combination vaccines with multiple different antigens. Safety aspects of consistence and residual material in the final product are important. So we have stabilizers in our vaccine products, we have preservatives, we have, as already mentioned, adjuvants. There may be residual material from the manufacturing process. We may have RNA, DNA sequences from cell lines, and so on. So what are we focusing on in vaccine safety surveillance? Especially the intensive investigation of rare adverse events becomes important. We need to have case definitions for the case ascertainment. Therefore, the Brighton case definitions have become so important. We have to do long-term follow-up in the post-marketing safe settings. Adverse events can affect the acceptability of immunization programs. Therefore, also intensive investigations become necessary. As I said, we have the age-relatedness of adverse events. We have safety aspects in different target groups. We have different methods of assessing causality of serious and rare adverse events. We have to look into the batch-relatedness of adverse events. We have to look into different methods for safety surveillance in the pre-licensure phase, in the development of our vaccines, as well as methods for safety surveillance in post-licensure, where we want to look into the rare events. And most important also is the vaccine risk communication. What methodology do we have to communicate vaccine risks? And vaccines, we have also cause-specific definitions. We have the so-called vaccine product-related reactions, where the adverse event following immunization, as we call it in vaccine safety, the AEFIs, are caused or participated by the vaccine itself when given correctly and due to one or more of the inherent properties of this vaccine. We also have the vaccine quality defect-related reactions, where the AEFIs are caused or participated by the vaccine that is due to one or more quality defects. And the quality defect are defined as any kind of deviation of the vaccine product as manufactured from its set of quality specifications, the so-called out of specs of the vaccine product, including the administration devices, meaning syringes with needles as provided by the manufacturer. We have the so-called immunization error-related reactions, where the AEFI is caused by, for instance, improper vaccine handling, providing a vaccine that has been out of the cold chain, prescribing changes, or inappropriate administration, where, for instance, a oral vaccine had been provided intramuscularly. And these, of course, are by nature preventable. We have also something special in vaccines, is the so-called immunization anxiety-related reactions. The AEFI arising from anxiety about the immunization, anticipating pain and fears related to the vaccine or its administration. I'm just referring to the syncopes seen in adolescents when vaccination is given in mass vaccination programs, for instance. Then, of course, we have the coincidental event, where the adverse event is caused by something other than the vaccine product, or immunization error, or immunization anxiety. I would like to draw your attention to some important specifics between vaccines and drugs. The vaccines are given electively and are given to healthy populations, mostly also at a vulnerable age, as we already have pointed out. The drugs are given to all populations that are sick, to ill populations, mostly adults, and they're rarely given electively. Vaccines have a complex composition, they're biological products. Still today, drugs are chemical products with many different drug classes. We do have more and more coming monoclonal antibodies that are also biologicals, but in general, drugs are really simple chemical products, simple chemical molecules. Vaccines are taking into account immunological considerations in addition to the pharmacological considerations, whereas with drugs we often look into the challenge, to de and re-challenge. These are important information for case assessment, whereas in drugs, de and re-challenge are not important for case assessment, or don't play not an important role as they do in the drug assessments. Vaccine may cause the illness they are meant to prevent, the vaccine-associated paralytic polio. But in drugs, we have to take into account many pharmacological considerations. Vaccines have a short duration of exposure. We give one dose at once, so we may give, second dose after some kind of a time period, depending on the nature of the vaccination. Drugs are given for a longer time period. They're rarely given only once or twice. Vaccines have a long time for response. However, many adverse events are reported for drugs over the long time period they are given. And minor events have an important impact as they may indicate a program error, whereas for drugs, minor adverse events are really important or considered to be important. So expertise is really needed for causality assessment in vaccines adverse events as compared to the causality assessment of drug adverse events. Now let me move on to the guideline on vaccine pharmacovigilance. The European Union had set out the guideline on good pharmacovigilance practice, the so-called product and population-specific consideration, vaccines for prophylaxis against infectious diseases. This guideline has become effective by the December 2013. And let me just give you an insight on what this guideline comprises. The objective of this guideline is to strengthen the conduct of pharmacovigilance for vaccines, but it does not replace the information provided in the other good vigilance practice modules. The module relevant to vaccine used in pre- and post-exposure prophylaxis of infectious diseases. And the module focuses on vaccine-specific aspects to be respected when designing and implementing pharmacovigilance activities for vaccines. So the module provides guidance specific for vaccines in relation to the pharmacovigilance process as described in some good vigilance practice modules. Specifically on Module 5, the Risk Management System, Module 7, the Periodic Safety Update Report module, Module 8, the Post-Authorization Safety Studies module, Module 9 on Signal Management, Module 15 on Safety Communication, and there is an additional module that provides guidance on batch recall and quarantine, where the legal basis is based on the GMP and GDP compliance, good manufacturing practice and good distribution practice. Now, let me go into these various modules I have just mentioned. Module 5, Risk Management Systems, what is specific for vaccines? In general, as I said, most aspects of the GPV Module 5 on risk management systems are applicable to vaccines. However, this GPVP1 adds some vaccine specificities. I have added them in red here below. Part 1 on the product overview, this is more extensive as we have to describe the product and all the product characteristics based on the production and manufacturing of the vaccine. In Part 2, that refers to the safety specification, we have to refer into the epidemiology of the disease and the target population. Is this now a pediatric vaccine? Is this a vaccine for the population from adult population or adolescent population? We have to refer to the non-clinical part of the safety specification on the preclinical development of our vaccine. We have to, of course, additionally provide information on populations that are not studied in the clinical trials. I'm just referring to pregnant female, breastfeeding females, immune-compromised subjects. These are all not studied or usually not studied in clinical trials. So this has to be defined here in the risk management system and how we consider to address this kind of risks. We have to also give additional EU requirements for safety specifications that may be requested when after discussions, scientific discussions with the European Union, with EMEA, there might be some additional requirements that have to be added for our vaccine. Then we have to be very explicit on the identified and potential risks of our vaccine, or what we have seen in our clinical development program. And the summary of safety concerns has to be very complete on what we consider to be of a concern for our vaccine being distributed or administered to a large population. In Part 3 on the pharmacovigilance plan, of course, we have different ways of routine pharmacovigilance activities, on different kinds of additional pharmacovigilance activities, on different kinds of post-authorization efficacy studies to make sure that our vaccine is still efficacious or effective when on the market, that the benefit-risk profile is still positive for our product. And of course, the risk minimization measures may look differently for vaccines as they would do for drugs. In Module 7 on the periodic safety update report, additionally, we have to provide some additional information on the PSURs for vaccine. We have to look into any potential impact on safety of changes in the manufacturing process. So we have to add there the specificities on changes in the manufacturing process. We have to evaluate the batch and age-related adverse reactions. We have to do analyses of adverse reactions for different doses or different vaccination schedules, vaccination regimens. Reports on vaccination failure, lack of efficacy, effectiveness, going to be very extensively discussed in the periodic safety update report. Therefore, we have to have a very thorough understanding on our vaccine effectiveness. Vaccination errors have to be added there. Vaccination anxiety-related reactions, such as syncope, have to be analyzed in the DSUR. Literature data relevant to similar vaccines, other vaccine components, different stabilizers, adjuvants especially, preservatives have to be analyzed. And an integrated benefit risk analysis using all available data has to be provided. Prevention of target disease, severity of symptoms, hospitalization, complications, effect of target disease on offspring in case of vaccination of pregnant women is an important part of the PSUR. The next slide shows you the Module 8 on post-authorization safety studies. So the objective methods and procedures for such past studies must be followed as described in our Module 8. The safety parameters in the past must be appropriate for the specific vaccine. There must be globally accepted standards for case definitions. So the Brighton case definition should be used whenever available. Vaccine registries may be used in vaccine safety with the respective caveats for active surveillance of rare adverse reactions. And non-clinical studies and experimental investigations have to be considered to address safety concerns, like virological, bacteriological, immunological experiments or other methods have to be taken into consideration. Module 9 goes into the signal management. And the signal management process is laid down in Module 9. That must be followed. But it also has to be relate to different fields of vaccines. So the evidence of reduced efficacy or effectiveness have to be looked into in the signal management process. Vaccine failure have to be looked into. The quality deviation with potential impact on safety, efficacy, or effectiveness, batch-related issues have to be looked into. Again, the Brighton case definition should be used whenever possible, and if there's not a Brighton case definition available for the adverse event of interest, we have to be very consistent on how we define our cases. What is important to know is that a single report of serious event only has to be processed as a signal in case there is a causal association to the vaccine. So not related adverse events are not necessarily to be processed as a signal. However, in clinical development, we will of course also still do that we will do signal management processes for not related adverse events that are of interest. They're the specificities of signal detection and mass vaccination program, where we have to look into these analyses observed versus expected. There are special considerations in vaccine signal detection when performing these kind of statistical disproportionality analyses. And we, of course, have to do the signal validation as described in the SIAMS WHO Working Group on Vaccine Pharmacovigilance. There's a specific section on signal validation on vaccines. Module 15 refers to the safety communication of vaccines, and as also here, the principles and guidance on safety communication planning and effectiveness evaluation is laid down in Module 15 and has to be followed. However, there are some vaccine-specific safety communication issues. They have to include information on avoiding errors in vaccine handling, administration, and reiterating precautions and warnings. We have to describe the benefits of the vaccines very explicitly. As I already pointed out at the very beginning, the benefits of vaccines are not seen immediately. We have to explain the risks for individuals and the population of a decrease in vaccination coverage, what that means to the population. We have to consider that risk perception may differ between the stakeholders. And we have to contain relevant background rates and exposure data when quantifying safety concerns. The communication planning must include preparedness for frequent public communication needs, and we also have to rely on the competent authorities that they must ensure appropriate communication with public and media and maintain a high level of transparency on how regulatory decisions were reached. I'm now coming to the end, and I would like to stop with the challenges in vaccine safety. There is still continued prevalence of unsafe injections and injection practices. There is mishandling of rumors and adverse events. Rumors have become legs. They go all around the world. There is lack of access to newer safe technologies such as autodisposable syringes, mainly in emerging countries. There's still inadequate surveillance of adverse events following immunization. And there is of big concern the growing anti-immunization movements, including these anti-vaccination websites. So the globalization and the internet had a great impact on misinformation, raising public concerns about harm from vaccines. The backup, I had actually provided you a slide on the new European pharmaco legislation, which you can have look into whenever questions come up from your side. Overall, I would like to thank you for your attendance, and goodbye.